Overview of analytes, amino acids. The basic structure of an amino acid is depicted here. So you can see it has an amino group, NH2, a carboxyl group, COOH, and then it has this central carbon and hydrogen known as the alpha carbon, and a side chain that's connected to it. This R group or side chain determines the identity of the amino acid. Metabolism, so essential amino acids must be supplied by the diet in the form of proteins. So when you consume this, the digestive tract will break down those proteins into the amino acid building blocks. Those amino acids are then absorbed into the bloodstream and from the small intestine, and then they go to the liver in order to be assembled into the proteins that we will need in our bodies. So amino acid requirements, there are essential, non-essential, and conditionally essential amino acids. You should be familiar with which ones are essential because these ones are extremely important to be supplied in the diet. So histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Conditionally essential means that these amino acids are required at certain times. So sometimes we can produce enough. They're required in small enough quantities that we have the ability to produce them under certain circumstances, but we want to make sure that they are supplied in the diet at other times. So we call them conditionally essential. Amino acidopathies refers to a group of inherited enzyme defects that inhibit the metabolism of certain amino acids. They exist either in activity of specific enzyme in metabolic pathway or in the membrane transport system for amino acids. Some states require screening for up to 26 amino acids. So down at the bottom here, I have the state of Wisconsin newborn screening website, so you can go see the different types of tests that are required in newborn screening. That's not just the amino acid tests, it's all of the tests that we test for, but many of them are amino acidopathies. There are over 100 diseases that result from this. They cause severe medical complications due to a buildup of toxic amino acids and byproducts of amino acid metabolism in the blood. So phenylketonuria is probably one of the more common ones, the ones that we have heard of a little bit more. Maple syrup urine disease is also another one that that might be familiar to you. The other ones are pretty rare. Tyrosinemia, alkaptonuria, isovaleric acidemia, homocystinuria, citrullinemia, arginosuccinic aciduria, and cystinuria. Urea refers to the presence of it in the urine, and then the stem of this, you can see kind of what amino acid it may be linked to. So phenyl can refer to phenylalanine, tyrose refers to tyrosine, isovaleric has to do with valine, like an isomer of valine, cysteine, homocystinuria, and so forth. So you can kind of deduce what these are related to. And this is not an extensive list of all amino acidopathies. These are just the more common ones. So amino acid analysis, blood samples should be drawn after at least a 6 to 8 hour fast. Sample is collected in a heparin tube and the plasma is removed promptly from the cells. You want to take care not to aspirate the pellet and the white cell later, and you don't want any hemolysis. If you think about it, if we do have hemolysis, then all of the amino acids that were loose inside of the cell will now be in the plasma. And we only want to evaluate the presence of the amino acids in the plasma. If we have hemolysis, we're going to have massive lysis of the cells, and every amino acid that was inside of the cell that was going to be used for protein production will now be in the plasma, and that's going to give us false results. Deproteinization within 30 minutes of sample collection. So we basically need to get all the protein out of there. Proteins over time will start to degrade and break down. So if we do have proteins in the plasma, we have an abundance of amino acids that can start to falsify our results. So we need to make sure that we get rid of the protein and leave just the plasma with the amino acids. Analysis is performed immediately, or the sample has to be frozen at -20 to -40. Urinary analysis is performed on random specimens for screening, and the method of choice for screening is thin layer chromatography.